Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. On behalf of the committee, we warmly welcome and sincerely appreciate the presence of our honored guests. From Faculty of Medicine, Universitas Indonesia, Head of the Master's Program in Biomedicine, Professor Dr. Wawaimuli Arazal, Masters of Biomedical Sciences, Ph.D. Secretaries of the Master Program in Biomedicine, Dr. Rani Wardani Hakim, Apoteker, Masters of Biomedical Sciences, Head of Department Faculty of Medicine Universitas Indonesia, Professors, all lecturers, and academic staff of the postgraduate program in Biomedicine Universitas Indonesia. From Institute of Biopharmaceutical and Health Engineering, Shenzhen International Graduate School, Tsinghua University. Vice Director of the Institute of Biopharmaceutical and Health Engineering, Shenzhen International Graduate School, Tsinghua University, Professor Xin-Hui Xing. Associate Professor Liu Xiao from Tsinghua University. Professors, all lecturers, and academic staff at Institute of Biopharmaceutical and Health Engineering, Shenzhen International Graduate School, Tsinghua University. Also, we warmly welcome to our beloved participants. Welcome to today's guest lecture, hosted by the Master Program in Biomedical Sciences Faculty of Medicine Universitas Indonesia, in collaboration with the Institute of Biopharmaceutical and Health Engineering, Shenzhen International Graduate School, Tsinghua University. Let's give warm applause for us. I am Sylvie, deeply honored to be your host for today guest lecture at the Faculty of Medicine Universitas Indonesia as a platform for sharing insights, strengthening academic partnerships, and fostering innovation in the field of biomedical research. It is truly an honor to have all of you here, both offline in iMeri Auditorium and online via Zoom, to join this insightful session. Today's event will feature a series of inspiring lectures delivered by outstanding experts from both Tsinghua University and Universitas Indonesia. We will explore exciting topics in biomedical sciences areas that represent the future of innovation in medicine and life sciences, including the first topic is synthetic biology in biomanufacturing of pharmaceuticals and nutraceuticals, to be delivered by Professor Xin-Hui Xing from Tsinghua University. Sorry, I just called. It's okay. The second is iPSC liver organoids and cellular immunotherapy for cancer, to be delivered by Associate Professor Dr. Radiana Dhewayani Antarianto, M.Biomed, Ph.D. from Faculty Medicine Universitas Indonesia. And the last topic is skin microbiome and aging, to be delivered by Associate Professor Liu Xiao from Tsinghua University. We hope that today's program will not only expand our scientific perspectives, but also strengthen collaboration and friendship between our institutions. Ladies and gentlemen, please rise for National Anthem Indonesia Raya. Thank you. Please be seated. And now, we would like to invite the head of the Master's Program in Biomedical Sciences to deliver the speech to Professor Dr. Wawaimuli Arozal, Master of Biomedical Sciences Ph.D. Please come to the stage.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Distinguished professor from Tsinghua Shenzhen International Graduate School, respective colleague from University of Indonesia, esteemed guests, and dear student, very good morning to all of you. It is truly an honor and a pleasure to welcome all of you to the Faculty of Medicine Universitas Indonesia. Today marks a special occasion, not only the signing of the Memorandum of Agreement for our double degree program between the Institute of Biopharmaceutical and Health Engineering Tsinghua Shenzhen International Graduate School and the Master Program in Biomedical Sciences Universitas Indonesia, but also the beginning of a two-day academic exchange filled with lectures, discussion, and student presentation. The next 2 days are the mean to strengthen the bond between our institution. Through the guest lecture, we will gain valuable insight from distinguished professor of both iBHE Tsinghua University and MPBS Faculty of Medicine Universitas Indonesia, who will share the expertise and perspective on recent advances in biomedical and biopharmaceutical sciences. At the same time, our students will have the opportunity to present their research, exchange ideas, and learn from one another, which will be an experience that truly gives the spirit of international collaboration and scientific curiosity. The double degree program is an important milestone in our shared vision to prepare future scientists who are global-minded, research-driven, and committed to improving human health. By combining the strength of Tsinghua University cutting-edge biopharmaceutical research and University of Indonesia solid foundation in biomedical science, this partnership offers our students an invaluable experience to grow in both academic excellence and cross-cultural understanding. On behalf of the Master Program in Biomedical Sciences, I would like to express my sincere gratitude to our partners in iBHE Tsinghua SIGS for their trust and collaboration, and to the faculty members, staff, and the students from both sides who have worked so hard to make this event possible. May this 2 days program not only mark the beginning of a fruitful double degree collaboration, but also inspire meaningful connection, innovative ideas, and a shared commitment to advancing science and education together. Thank you very much, and I wish everyone an enjoyable and meaningful event. Thank you very much.
Thank you very much for Professor Muli for the inspiring speech. Ladies and gentlemen, we are now about to begin our session. The guest lecture topics that will be moderated by Professor Dr. Melva Louisa, Master of Biomedical Sciences from the Faculty of Medicine Universitas Indonesia. Allow me to briefly introduce our moderator. Professor Melva Louisa has been an academic staff member in the Department of Pharmacology and Therapeutics at the Faculty of Medicine Universitas Indonesia, teaching pharmacology since 2006. Before her appointment at FMUI, she worked as a trainer in medical sciences for pharmaceutical companies, training medical representatives. Currently, she serves as the research coordinator for her department and is part of the academic quality assurance team for the doctoral program in biomedical sciences. She received her Ph.D. in biomedical sciences from the Faculty of Medicine Universitas Indonesia in 2012. In June 2023, she was appointed a professor of pharmacology and therapeutics. Her background in pharmacology and research-related activities started in 2006, when she served as clinical research monitor at the Clinical Research Supporting Unit at FMUI until 2018. She has also published many articles in clinical research. Together with her colleagues in the department, she works with the Indonesian FDA on the new drug evaluation process. Without further ado, please welcome Professor Melva to moderate this session.
So thank you, Master of Ceremony. It's an honor for me to chair this session, this guest lecture. So today we will have three distinguished speakers. And then the first one, it's a great honor to welcome Professor Xin-Hui Xing, one of the world's leading experts in biopharmaceutical and health engineering. And Professor Xing earned his PhD from the Tokyo Institute of Technology, and today he serves as the Executive Dean at the Shenzhen International Graduate School. His research covers the enzyme engineering, synthetic biotechnology, bioactive peptides, and carbohydrate drug technology. And he has published over 350 publications and recognized as one of the world's top 2% scientists. And this morning, Professor Xing will speak of synthetic biology and AI-empowered biomanufacturing of pharmaceuticals and nutraceuticals, a topic that promises to open new perspective in science, technology, and global health. So please join me in warmly welcoming Professor Xin-Hui Xing.
Thank you very much, Professor. Is there any? Yeah, pointer, yeah. So it's I said the delegation of Tsinghua from Tsinghua Shenzhen International Graduate School. I would like to express my sincere thanks to all of the professors from University of Indonesia. The Faculty of the Medicine. I think especially I would like to give my sincere thanks to Professor Arozao. So later I will show you what we have met last time in June in Shenzhen. Shenzhen is a very, very beautiful city and international city, and also the inclusive city. So we always say that if you come to Shenzhen, you will become Shenzhenese. Last time I told Professor Arozao. So today, I think the first part, I will take several minutes to introduce the Institute of Biopharmaceutical and Health Engineering, Tsinghua Shenzhen International Graduate School. In short, the Tsinghua SIGS and also iBHE. Sorry. So Tsinghua has two campuses in China. The first one, the main campus is in Beijing. So Shenzhen campus is only one campus outside of Beijing in China, but now it's in building next international campus in Middle Asia, the Middle East. Anyway, I think that's a future story. So in China, only the two campuses. In Shenzhen, we are taking the education of the graduate school, but in main campus of Beijing, we have the undergraduate education and also the graduate education. Sorry. So I think yesterday I just checked the distance from China, from Beijing to Jakarta, from Shenzhen to Jakarta using DeepSeek. That's AI tool in China. It's very, very convenient, very powerful. We can get many the precise data by using the DeepSeek. Something like the ChatGPT, but more convenient than ChatGPT, I feel. So from Beijing to Jakarta, around the 5,000 kilometers. From Shenzhen to Jakarta, only around 3,000 kilometers. So that means we are very close. Just 4.5 hours, we can reach the Jakarta. So I feel really Asia is very close. So we can become the best friend. So what's the? So this is I think the statistical data, just also using the DeepSeek. I think this is quite the new data. So firstly, the trade and the investment between China and Indonesia, I think now is very huge. It's 150 billion in 2024. Also, we are increasing the people-to-people exchange in different areas, including our scholars exchange. And for China and Indonesia, the cooperation and cooperation and collaboration, I think the first one is the university industry collaboration and the talent development. Like we are discussing the exchanging the research activity and also future the double degree or dual degree education program as promoted by Professor Liu Xiao and also Professor Arzola. So also we have many, the new future technology, the cooperation, including the energy and also green industry. That's a green economy. So also I think you know well, China and Indonesia has jointly they built a very good the high-speed railway system, and also there's many political dialogue, the activities. I think that means we have huge opportunities to collaborate together in not only education but also in the technology, the innovation collaboration. So this is the slide I just learned yesterday. Sorry. Okay, I think between iBHE and the Faculty of Medicine University of Indonesia, FMUI, so we have, from this year, we have several events, including the online meeting, and also in June 26 to 28, we invited the Professor Arzola and Professor Rani to visit Shenzhen International Graduate School, and also Professor Arzola gave a very exciting lecture in our the health engineering forum. So then I just move to the introduction, brief introduction of our iBHE. So this is the SIGS situation, Tsinghua SIGS. So we have 6+1 same area due to time limitation. The first one is our Institute of Biopharmaceutical and Health Engineering. And also the other one, the energy, the environmental ecology, data science, and also ocean engineering, the the future habitat, and also the material institute, plus the innovative management. Because 70% of the students will be educated now, are being educated in Shenzhen. So we have to teach students with their innovative the management, the skills, and also the knowledge. So that's I think the Tsinghua in Shenzhen is different from the main campus of Beijing. In Shenzhen we are focusing on the interdisciplinary research and also engineering. They are based the problem solving their education and the research. So currently in Shenzhen, we have around 6,000 graduate students. The 2/3 is their master program, and 1/3 is the Ph.D. program. I think this is the rough data. And for our iBHE, sorry, this is slow. So we are challenging the global health, technology, and also the translation research for the preventive and the healthcare and also the disease control. So I think as I mentioned, we using the chemical engineering approach by interdisciplinary joint the research with the life science and medicine, and also the material and biomaterial, and also nano and biomaterial, and also AI data science. So currently we have 3 key research directions. The first one is cell engineering, and also the evidence-based health engineering, and also the pharmaceutical engineering, including the vaccine engineering. I think in this sense, we have a very, very close collaboration potential with Faculty of Medicine of University of Indonesia. So you are very close to the clinical research and also medicine research. I think in this sense, we can produce many biopharmaceuticals and also healthcare products. Then we can have the wide angle for doing the collaboration between the medicine and the engineering. I think that's a very, very important research future. So currently we have 30 PIs, but by 2030 to 2035, our PI will be increased to 60 and 70. So that means we can also have a big potential for the faculty exchange. I really hope your outstanding young faculty, if you have interest, you can come to Shenzhen to do the Tsinghua PI in Shenzhen. I think that's also, Tsinghua Shenzhen has a very good salary system. Then currently we have around 500 students. Later, Professor Liu Xiao will give the details. But by after 2030, our student number will be increased more than 1,000. At that time, total graduate student number of Tsinghua SIGS will be over 10,000. So that's a very, very big capacity. Among all the students, we have a very, very international KPI. So after the 2030, so the faculty and the students with the foreign nationality will be increased to 30%. So that's also, we hope that many exciting students from the University of Indonesia and also from the other countries can come to Shenzhen, because Shenzhen is very international. So we use bilateral languages, Chinese and English. In Shenzhen, it's a same language. So we have currently we have more than 60% of the courses, teaching courses. We are giving the lectures in English. So that's don't worry if you don't understand Chinese, but of course welcome you to learn Chinese. So this is the basic information. So we also in China we are also promoting the so-called organized key research initiatives. So we are focusing on the transforming discovery to manufacturing. So based on the three research directions, and also the problem, the orientation, engineering research approach. So we set up the the joint research areas for the translation. The first one is the medical and health, the instrumentation, and also big data engineering. Based on this platform, we are doing the green biomanufacturing of the pharmaceuticals, and also we are doing the green biomanufacturing of the healthcare product, the Nutraceuticals. So that means we will combine the AI technology, and also the omic technology, and also their clinical data. Then we can do a more precision medicine and precision healthcare, the product, the manufacturing technology, and the production technology. I think that's our picture. So last time the Professor Randy and the Professor Arizona also they visit our research platform. We have a very, very good shared innovation and research platform, including teaching platform, research platform. We have including our 3 research areas, and also we have the BSL2 level laboratory, and also we have animal facilities. So this is, I think, the brief information. Sorry. So I think maybe we are also actively promoting the international collaboration by developing global collaboration and to establish the international joint training program. Like today, I think we will sign the MOA with Faculty of Medicine, University of Indonesia. So we have done a lot of the agreement with different universities in different countries. So in future, I think we can put the University of Indonesia into this rank. So also we have launched the iBHE founded the international journal. So under the support of the Tsinghua Press. So now we have published the first 10 papers with a very, very high quality. So future, this journal is health engineering as a very, very interdisciplinary research. The topics can be included. So I really hope in future we can also collaborate with the Faculty of Medicine, Indonesia University. So then I just take around 15 minutes to talk about what I'm doing in my group. So in total, I'm engineering background. So we are doing the biochemical engineering, like introduced by Chair. So in total, I think currently it's one is synthetic biology. Synthetic biology is a platform for doing the engineer life or engineering the life science. So another key technology is the AI. So sorry, skip too quickly. So we are doing these two technologies empowered the biomanufacturing of pharmaceutical and nutraceutical. So my purpose, my research, key research interest, how can we promote the function discovery to the function manufacturing or function engineering? So this is, I think, engineering science should give the answer. So this is my representative projects in my group. The first one, we have actively developed the enabling biobreeding technology and instruments for advancing the synthetic biology and biomanufacturing research and also translation. Based on these tools, so now we are doing the development of the next generation heparin drugs for treating the autoimmune diseases, including UC and RA. Then today, due to time limitation, I will just pick up the recent research area we call the AI-added the mining technology for the bioactive peptide for discovery and also the manufacturing of peptide drugs and also peptide the nutraceuticals. That's healthcare, the functional food. So then I think I'm not sure you are familiar with the peptide, bioactive peptide. As fixed bioactive peptides are from the natural protein sources, of course. So including the plants, animals, and the bacteria, the microbes. So based on the past 100 years research, now people are well known about the health benefit of bioactive peptides. They are from our head to our feet. So I think there are many clear targets which the bioactive peptide can combine, then can control their chronic disease or their important disease. So that means bioactive peptides have the increased the application in pharmaceutical and also the nutraceuticals. So, but so far, bioactive peptides, protein resources which have been explored in the world are very limited. So we have a very limited bioactive peptides with non-structure and also function. And also in many cases, I think they also in Indonesia, the genome or the proteome information for many natural protein resources are still unknown. So how can we solve the problem? So currently, all the bioactive peptides production and also their mining, their approach based on the top-down, we call the top-down the method, including the fermentation, including the protease, the degradation, Then by this technology, we have to do the separation, purification, and the function, the exploration, and the screening. So that will take the, become the complex process, and also will consume the very large, huge labor time and cost. So that's, I think, the big problem. So what's the future, the new strategy? We are thinking if we can use the AI and the computation, combine the technology, then maybe we can develop the effective mining, the method for the functional peptide, from the different, the genome and protein and the species, then to accelerate the developing the speed of the bioactive peptide from nature. So that's, I think, then I will go to the quickly. So this is a cartoon summary of what we have done in the past 8 years. So we established the AI plus computation driving the systematic discovery of the bioactive peptide. So the key is they are for the unknown genome sequence, also protein sequence. So we use the transcriptomics, the analysis method, combining with the proteomics, the conformation. Then we can know the sequence of the protein in different species. Then after that, we can use the AI and also the molecular docking method by binding with the target related to the different diseases or the chronic diseases. Then we can construct the very, very huge the peptide library and also functional bioactive peptide library. So by that way, we can get the predicted the bioactivity. Then we can synthesize highly active bioactive peptides. Then we can confirm their function and mechanism. Then we can feed back to the production with predetermined their powerful proteases. Then we can get the high activity, the high content of the peptide the mixture. If we use the peptide mixture for the functional foods. So this is, I think, basically the research, the strategy in my group. So I just give one example. So we have done a lot of the research actually in the past several years. Right? So this is just an example. So if we look at the diabetes, so there are a lot of the targets or the receptors from head to the muscle. So they are currently all the anti-diabetes drug development is based on the intestinal. One is the GLP-1, the receptor, the agonist. Another one potential the target is the DP4 inhibitors. So I think later I will just give a very brief introduction. So the strategy aiming at the multi-targets for diabetic treatment and prevention, I think this strategy is very, very important. Not only one receptor or one target. So so currently, I think the most active research are focusing on GLP-1, the receptor. I think that one you know well. So it's a agonist. Now it's very actively being developed. So they are huge. The market, the drug I think they are semi-glutide. So that one, the last year, I think the world market is more than around the 3 million, 30 billion the US dollar. I think it's a quite the big market. But on the other hand, the DP4 is also very important. DP4, if DP4 activity becomes higher, DP4 will degrade the GLP-1. GLP-1 is very essential, the peptide, which can increase the insulin secretion. So if the GLP-1 is degraded, the lifetime is short, then the insulin will be not enough. Then the blood sugar will be increased. So in another case, for the diabetes, always the DP4 activity is increased. So if we can inhibit the DP4, the activity, maybe it will be more safe for the anti-diabetes drug development. But so far, except the small molecular DP4 inhibitors, there are very few peptide peptide inhibitory targeting DP4 have come into the commercial stage. So that means we have a very huge space to develop the DP4 inhibiting the bioactive peptide as a functional food or the drug from the viewpoint of the safety and also the efficacy. I think this is the story of my research. Sorry. So we choose we call the homology of the food and medicine, the plants. That called the industry hemp. Industry hemp is in Chinese the huoma. So the hemp seed have been used for more than 2,000 years as a healthy and longevity food. So the people in China eating this every day, they eat this one, so they can feel their long life. But the protein is, of course, is a very high quality protein. But very few research on the hemp seed peptide and biomanufacturing have been they are studied. So we established the AI, as I mentioned, the AI and also competition, and also the molecular docking. They combined the technology approach. Then finally, we established a very huge. They are they are around the 180,000 the peptide library, and among this the bioactive library. So we found the DP4 inhibition, ACE the inhibition. That's for pressure control, and also other inflammation activity, anti-inflammation activities, very wide the potential activities. So then, because we are interested in the development of DP4 inhibition bioactive peptide, so we choose the high activity, potential the peptide with the DP4 inhibition, 69 types of the peptide with a high activity, DP4 activity. So then we found that 34. The IC50 lower than 1 millimolar is very high. And also one peptide, the IC50 lower than the 1 micromolar. That means the druggability is quite good. So I think this also the very interesting the discovery. Then I will go to the quickly, sorry, maybe I consume time. So this is just to show us that we choose a very good enzyme which can generate the very high DP4 inhibition activity, and also the highest active GLP-1, the level, and then the significant glucose homeostasis. Then based on the AI and also the prediction technology, intensity affinity detection, the combined detection, we got the 24 potential peptides. Then from 24 peptides, we screened the 5 peptides for with the potential analysis. Then from 7, we identified one strong and very most active the functional peptide, the tetrapeptide VMP, the very short peptide. So then we for the further analysis, they just published the iBHE last 2 months ago. So this is an analysis of the mechanism of the DP4 inhibition by this peptide. I will not go to detail. It's a competitive inhibition, and also with a very strong binding affinity with DP4, and through the hydrogen, the interaction, the binding, and also the ion binding, and also the hydrophobic interaction, etc. I think we identified all the binding sites. So then also we did the VMP in vivo, the pharmacokinetic analysis. So this one is the 4 tetrapeptide very stable in the stomach for to resistance to the gastric digestion, and also around the 11% they detect in the blood. That means the function to to the DB4 and also GLP-1, and then also the liver, the lipid, the metabolism. And also the very interesting, around the 74% of the VMP remained in the intestinal content. So that also, later I'll show you that's what this one is doing in the intestinal. So this is also very interesting, I think, the data. Okay. So we also systematically, we analyzed the VMP inhibition, inhibition of the DB4, then to promote the insulin secretion and reduce blood glucose metabolism. So this also, I will not go to detail. And then we, then we analyze, this is very interesting. So VMP improves the gut barrier function by promoting the AKK speed, the abundance. This is a very, very useful probiotic, the inert probiotic, the strain in our gut. So among all the microbiota, the structure change, this one I think is specifically increased. Then we analyzed why the VMP can regulate the AKK strain in our intestine. So we found that this one really can, combining with the fecal, the FMT, the tools, technique. Then we mimic we found that only VMP, the addition can promote the AKK strain, then to increase the very different phenotype, including the plasma TG, the lipid the level. I think this that means also can control the weight. So finally, we found this mechanism of the VMP promotion for the AKK abundance in the intestine. That means that by the upregulated expression of this gene related to the increased synthesis of the peptidoglycan. That's one very important for the AKK strain function for increase the mucosa barrier and also to increase the anti-inflammation and also increase the glucose, the metabolism. I think also this one is the very important data. Sorry, very slow. Okay, this is a summary of our research. I think there's with DI and molecular docking and also the AI prediction. So we get finally we found one type of peptide VMP. As I mentioned, the one way is for the glucose metabolism, the control through the DP4 inhibition and also GLP-1 increase. On another hand, also that one is the higher the percentage remain in the intestine, in the gut. Then they will specifically increase their AKK strain, the abundance, the growth. So by these two key pathways, then it decreases the blood glucose, decrease the body weight by control the lipid, the synthesis. I think this VMP also has a very good drug ability. So now we are doing this synthesis and also the drug development, the process. So So we also, based on our research technology development, so now we have built up the production plant, the plant in China, in prefecture, in the prefecture, with production of the Hep C. So then this one is just based on the enzymatic biomanufacturing of the active peptide. So for the nutraceutical the application. So we have developed many products. And another one is we are developing the synthetic biomanufacturing tools for this peptide for pharmaceutical development. So we're using the yeast, the petaries as a host. Then we're doing one is genetic element discovery and the AI-based modeling and the chassis construction. Then we're doing the absolute screening. Now we have reached a very, very good strain development. with a high productivity of the VMP and also other bioactive peptide with a potential application as a pharmaceutical peptide pharmaceutical development. I hope in future we can further discuss in detail for this part. So finally, so I should give my success in Chinese, but since all the collaborators also collaborated the worldwide, and also for the students in my group, especially for and also for the funding support. And thank you very much. Okay.
So thank you, Professor Xing, for such an important topics on synthetic peptides and AI biomanufacturing. And it is my pleasure also to welcome over 90 participants online, aside from us here. And then you can also drop your comments and questions on the chat box. So we will present it on the question and answer time. Okay. And then our next speaker is our own beloved Associate Professor, Dr. Radiana Dhewayani Antarianto. Yeah, she is from the Department of Histology, Faculty of Medicine, Faculty of Medicine Universitas Indonesia. She currently coordinates the stem cell concentration in the Master Program of Biomedical Science, and she also leads the Stem Cell and Tissue Engineering Research Cluster at iMERI UI. And we are very proud that she is an internationally trained with Ph.D. in regenerative science from Hanover Biomedical Research School in Germany, and has pursued advanced training in tissue engineering at the National University of Singapore, and have a research fellowship at Osaka University, Japan. Her research focused on the stem cell iPSC-derived liver organoids and also cellular immunotherapy for cancer. Today, she will share her insights on the iPSC liver organoids and cellular immunotherapy for cancer, a topic at the frontier of regenerative medicine and oncology. Please welcome Associate Professor Dr. Radiana Dhewayani Antarianto.
Thank you, Prof. Melva, for that very kind and nice introduction. Prof. Muli as well. I'd like to thank the head of Master Program Biomedical Science, Prof. Muli. I'm just following her direction because actually I wanted to learn more from our distinguished guest today from Tsinghua Shenzhen International Graduate School. But yeah, following the footstep of Prof. Muli direction, so I think we will present what has been done in our stem cell and tissue engineering research cluster. Can I have the slide, please? Okay, so today's topic is about iPSC liver organoid and cellular immunotherapy for cancer. Next slide. I want to show you where we do most of our work. So currently in Faculty of Medicine Universitas Indonesia, we have two facilities. The first one is in this building, 8th floor, research tower. So it's the Stem Cell and Tissue Engineering Research Cluster. This is where we teach our students, we give training as well for graduate student, undergraduate medical student, as well as our master and PhD students in stem cell or tissue engineering related topics. So every research that is done in iMERI, it's preclinical research, in vitro study, in vivo study to have proof of concept in the animal models, and we have the second facility. So this second facility is located at Cipto Mangunkusumo National Referral Hospital. And the product, the stem cell product, secretome, metabolite product of stem cells that is generated in this facility, it's under cGMP standard, already certified by Indonesian FDA to produce stem cell, secretome, and metabolite products, and to go to clinical trials with the patients. So we have these two facilities. Next slide, please. Okay, so moving to our first topic about liver organoids. Actually, our focus on liver regenerative medicine comes from being inspired by the liver as the vital organ in our body. The function is for protein synthesis. We check a lot of albumin markers as well as metabolism or drug biotransformation, which we will look into the cytochrome P450 enzyme and also the detoxification function of the liver. When there is a liver damage, whether it's through hepatitis B or C, drug-induced, toxicity, genetic disease, or drug abuse, actually the liver is one of the most abundantly regenerative organs. So the liver the hepatocytes can proliferate and they can overcome some of the liver damage. However, when a certain threshold or a certain level of necrosis and apoptosis of the liver cells occurs, then there is no turning back. The fibrosis of the liver can occur, it will eventually progress into cirrhosis and end it with liver failure. Of course, in Indonesia, it has been reported by WHO in 2015 that the liver cirrhosis is one of the 10 causes of death in Indonesia, with a mortality rate is quite high in Indonesia. For men, it's 52.7 per 100,000 men. In female, it's 16.6 to 100,000 female. The only available option for end-stage liver failure would be liver transplantation. Of course, until now, I would also happily like to share the story that in Indonesia, Cipto Mangunkusumo National Referral Hospital is also center for liver transplantation. So up to now, 100 baby with biliary atresia have received liver transplantation successfully. But of course, there is lack of compatible donor organ. It takes high expenses, and the patient needs to undergo long-term immunosuppression. Other alternative, because the liver, the parenchyma of the liver, the predominant cells, functional cells is hepatocyte, you can just transplant the hepatocyte. The problem with the hepatocyte transplantation is that once you take out and isolate hepatocyte from the liver and try to culture it in vitro in the lab, the hepatocyte cannot expand. They underwent proliferation arrest and then they just died in the flask. So we think about stem cell transplantation. Direct stem cell transplantation has been occurring all over the world in several research centers. There is a downside to it. Thrombosis of the liver and other minor or capillaries, as well as the report of tumorigenesis. So stem cell transplantation might not be the answer. So what we are thinking about is making a bio-artificial liver. Next slide. So initially, we started with the question in 2015. This question is managed by two of our master alumni from Biomedical Science, Universitas Indonesia. We asked the question, how or what is the cellular mechanism of stem cells to inhibit liver fibrosis? So we tackle this question by co-culturing three-dimensional of the stem cells. As you can see on the left-hand side, this is isolating the stem cells directly from the bone marrow of the rats. And the second one is using isolation CD34 markers, umbilical cord blood stem cells, and we co-culture it with the prominent cells that plays a role in fibrogenesis of the liver. The cell is hepatic stellate cells. Initially, into 2015, we have not gained the cell line of hepatic stellate, so we also do primary isolation from the liver for the hepatic stellate cells. But in the next alumni, we already have hepatic stellate cell lines of LX2. So what we are looking at the moment is the result. I cannot, due to the time constriction, I cannot share you all the figures, but you are welcome to look into our initial publication. So in here, we show that using co-culture of primary bone marrow-derived MSCs, there is an inhibition in the activity of the LX2 or the hepatic stellate cell, as shown here. The deposition of the Mason trichrome stain area, which corresponds to the fibrosis area of the liver, decreases. And in here, with CD34 hematopoietic stem cell umbilical cord blood, we also show that co-culturing the LX2 hepatic stellate cells with umbilical cord blood CD34 are able to inhibit the activity of the stellate cells, and they reduce the deposition of collagen as extracellular matrix. So we saw a very small volume in comparison to the monoculture of the hepatic stellate cells. Next slide. Then we asked the question, how to form liver organoids. So we have made hanging drop methods using two types of one hepatic stellate cells and two stem cells. So we asked the question, if we want to make liver organoids using primary hepatocytes from the rats, at what ratio, cellular component ratio, can it recapitulate the cellular structure of the liver? So we optimize the cellular concentration between 4 cellular components, primary hepatocytes directly isolated from the rat liver, and then 2 types of stem cells, the umbilical cord mesenchymal stem cells and the CD34 hematopoietic stem cells, as well as the hepatic stellate cells as the supporting cells. So eventually, after optimization, we noticed that with the cellular ratio of 50% primary hepatocyte, 10% LX2 cell, 20% of mesenchymal stem cells, and 20% of hematopoietic stem cells, you get a competitive advantage of viability of the hepatocyte up to 14 days in culture. While if you are only culturing the primary hepatocyte without the other cellular combination, it can only sustain the primary hepatocyte viability until day 7. What other type of things that we need to consider when we want to culture organoids is about the formula of the medium, of the culture medium. So we also make several optimization of the medium, and after the optimization, we found that the medium to cultivate liver organoids, the basal medium is Williams E supplemented with platelet lysate, insulin, transferrin, selenium, and dexamethasone. And we can show up to 14 days, not only structural composition of the liver organoid, but also the functionality as measured by molecular analysis as well as histology analysis. Next slide. So there is a problem with our liver organoids. 2 of the function of hepatocyte, which is to secrete urea and bilirubin, somehow diminish after 7 days in culture. So what we think happens is that when you make liver organoid only comprising of those cellular components, only the cell at the surface of the 3D co-culture can secrete urea bilirubin, and this will eventually end it. What happened inside, they got stuck. They cannot secrete the urea bilirubin accordingly. So our idea is to give space, to give extracellular space among the cellular components. So we wanted to make, coming from histology background, the extracellular matrix that is best, usually given by nature. That's why for making the extracellular matrix of the liver, we assign a master alumni again for, from Biomedical Science, Faculty of Medicine Universitas Indonesia, and she did this work. She was able to make a decellularized native liver scaffold using a very simple tools. Only 1 cc of syringe with a fixated by like cable wire. So we fix this syringe, and then we can precisely make or make the multiple site syringe injection. This is what happened when you decellularize in one side. This is when it's 2-site injection. This is with more than 3-site injection. And after 4 to 7 sites of injection, you get this really ghost membrane liver cubes, only containing extracellular matrix of the liver, as well as some vessels. We cannot identify it first, but we will later on. These vessels are actually coming from blood vessels of the liver or bile duct of the liver. Okay, so we have now the spacer, and this is also showing scanning electron microphotography to show the honeycomb appearance or pore size that is very naturally made and can hold the hepatocytes when we see the stem cells later on. Next slide. So, with those two technologies in mind, we have the capacity to have the cellular ratio, cellular component ratio. We have the capacity to also make a spacer between the cells. Now we move on into, can we make liver organoids from stem cells? Or because the primary hepatocyte, they have a very short lifespan in culture. So we turn into stem cells, and we use these two sources of stem cells. One is iPSC, and the other one is from mesenchymal stem cells. And the decellularized native liver scaffold was again generated by different alumni, but still having the same result, so the protocol is reproducible. And then we see this decellularized native liver scaffold with iPSC or mesenchymal stem cells, and we check for differentiation. So we are quite lucky because in 2019 there has been this protocol to differentiate hepatocyte directly from induced pluripotent stem cells in 21 days, and the differentiation protocol is actually GMP grade. So everything that we use here can be translated further to the patient later on. So using the protocol by Blackford, we modify a couple of cytokines or growth factors as well. We show that in 7 days of differentiation as well as 21 days of differentiation, we can still find some cells attaching to the surface of the scaffold. Okay, this is stained with routine hematoxylin eosin staining, and this is stained with Mazon trichrome. But what is interesting is that when you use mesenchymal stem cell as the source of stem cell in liver organoid, you get an extra, more abundant deposition of collagen. This can be good news, but this can also be bad news because we don't want to have more fibrosis in the liver organoids, okay? So with iPSC, it's okay. With the iPSC, you can still get the hepatocytes, although the number is fewer than the MSCs, and also the functionality is lower. However, with the maturation stage, the iPSC-derived hepatocyte in our liver organoid is higher. Okay, next slide. So, now we move on to the translational research. So our research question is, what happened when we transplanted this iPSC-based liver organoids to an animal model of cirrhosis, of liver cirrhosis? So the rabbit, we use rabbit. The rabbit was performed bile duct ligation surgery to make an obstructive biliary obstruction of the liver, and thus causing cirrhosis of the liver. 2 weeks post bile duct ligation, we perform another surgery to transplant the iPSC-based liver organoids in the rabbits. And as you can see, the transplantation is ectopic. It is transplanted in the peritoneum, omentum maius of the rabbits. And after the transplantation, it got sutured back, and then we monitor or follow up the viability of the rabbits as well as the functionality of the liver. I'm not showing you the positive result. Okay, so the positive result is that transplanted liver organoids can sustain the life of the liver cirrhosis rabbit for up to 7 days. When you expand this time period into 35 days, then you start to see progression of the liver cirrhosis, diminishing liver functions, and eventually death of the animals, okay? But there is a statistically significant improvement of the liver after transplantation for short term. When we talk short term, it's 7 days rabbit viability or survival. So this is the negative result. Then we ask the question, why is our transplanted liver organoids cannot sustain the life of the rabbit over 7 days? And indeed, these are the retrieved transplanted liver organoids. And as you can see, we have only bile duct ligated, so it's without liver transplantation. This is sham operated, also without liver organoid transplantation. And these 4 are actually coming from transplanted liver organoids in long term and in short term. What we are looking at is the infiltrates of the B cells and T cells. So because this is xenograft transplantation, our original stem cells, hepatic stellate cells, mesenchymal stem cells, and CD34 hematopoietic stem cells, all coming from human, it does elicit immune response in the rabbit because we are not giving the rabbit some immunosuppressive drugs. Okay, so the rabbits are highly immunocompetent, and it is showing here that the infiltrates are coming more and more when we took out this transplanted iPSC organoid in long term, as well as the viability of the cells decreases. So the cells get attacked by the immune system of the rabbit. Next slide. Okay, so having that in mind gives us a new perspective. Maybe we don't need to transplant the liver organoids. We need to have it still outside the body of the organism, but it can also maintain the survival of the organism. So this is a video. I hope it can play. I want to show you the video. So we place the organoids outside in a bioreactors and connect it to the circulation of the bile duct ligated rabbits. Okay, so these are our team, some hepatobiliary doctors, internal medicine. Can you make the sound? I hope that. Okay.
Mulai percobaan
Okay, so
untuk menciptakan bio
so I want to show you. So these are the bioreactors, and we place the iPSC liver organoids in the bioreactors. Now we have 4 liver organoids. This is the perfusion, to perfuse all the blood. And the blue one is actually only a temperature regulator. So it keeps the blood in 37 degrees. And the vascular surgeon, so this operation is done by a vascular surgeon. He make access from the femoral artery of the bile duct ligated rabbit, 2 weeks post bile duct, so it's already having some cirrhosis. And the entrance of the blood is to the vein in the ear of the rabbit. Okay. So I will show you what is the next challenge from our bioartificial liver. So we noticed previously our prototype is only having a perfusion rate of 2 milli per minute. But this one is 19 milli per minute, the perfusion rate. So it's like 9 to 10 times higher. Second, the scaffold or our organoid just goes out from the bioreactors, entering the perfusion, entering the IV line, although the vascular surgeon are able to disconnect and take out the iPSC organoids, but this is not supposed to happen. Okay, next slide. So what we did is that I want to show you, this is the original. So when we did the prototype of Shinta or the bioartificial liver in 2021, we only use simple perfusion bioreactors. This is the milli bioreactors. The organoid is in here, and this is the medium. And we only perfuse with the rate of 2 milli per minute. When it comes to the animal, there is viscosity of the blood and higher perfusion rate. And that just causes the scaffold to step away from the milli bioreactors. So this is the 3rd generation of Shinta, where the engineers have fixed the milli bioreactors. Now it's in a streamlined condition. And this circuit can hold the perfusion rate up to 19 milli per minute. So, yeah, next generation of Shinta is underway. So we're thinking about giving blood vessels to the liver organoids. And this will be the upcoming project of Salisu, our master program student, international master program student, as well as another doctoral student. We are also looking at Eko. Hi, Eko. So he's our doctoral student. He's fixing and try to avoid when the scaffold get thrusted into the circulation, then some cells will be dislodged or detached from the scaffold. That's why ECHO will be working with hybrid scaffold with some electrofiber nanospinning surface modified scaffold that can enhance the attachment of the iPSC-based liver organoids. Okay, so moving to the second topic. Next slide. Yes, okay. So now moving to the second topic, this time, this is actually one of IMERI's research challenge, growing up healthy. We know that cancer is one of the most devastating condition affecting the elderly, and cellular-based immunotherapy has been established. I think in China, you have so many alternatives for CAR T-cell therapy. Some are homegrown or developed in China itself, but in Indonesia, we don't have CAR T therapy yet. So there are a couple of cellular immunotherapy modalities. It's tumor infiltrating lymphocytes, so the Tregs that can regulate the microenvironment of the cancers are isolated from the blood of the patient and then expanded in the lab and then given back or reinfused to the patient. The second is engineered T-cell receptor therapy. So we see here that we isolate the T cells and then genetically modify the T cells to express certain type of T cell receptors, expand it and infuse back to the patient. So the CAR T cell therapy is the one that has been approved by FDA and has been used in several hematology malignancy in Japan, China, USA, Singapore, Malaysia. Unfortunately, it's not here yet, but we are looking forward to build the capacity to do this CAR T cell therapy. But between these three modalities, there is a problem. With the T cell therapy, you have a higher amount of hyperinflammatory state, and sometimes this patient just don't, there is no clinical improvement in the patient from the T cell therapy. So nowadays, the CAR NK cells are the one that is being investigated further in clinical trials. So this is PSA, prostate-specific antigen CAR NK cells. And there has been many more publications by using natural killer cells. So the difference when you use natural killer instead of the T cells is that natural killer cells have so many more surface receptors, activating and inhibitory receptors, and they work by signal resultant from these receptors. So NK cells are the one that is investigated further in our research cluster. Next. So this is our initial work in 2019. We try to avoid a genetic approach on inducing the capacity or the vitality of the NK cells. But we know that the NK cells can get activated by giving proper signal. So the proper signaling, after some literature search, can be given from exosomes. Exosomes are nanoparticles naturally released by cells. It can also be signaling molecules inside cancer microenvironments. So we induce the NK cells using exosomes. Initially, we try to characterize what is the difference between exosomes coming from cancer microenvironment and non-cancer microenvironment. And we notice only with simple SDS page, a couple of bands missing in the cancer microenvironment exosomes. So everything that we want to, add it or induce, we have to add from the non-cancer microenvironment. That's how we come up with the exosome-induced NK cells. Initially, we are collaborating with obstetric and gynecology, gynecology clinicians, Dr. Anggi. She provides us with a lot of ovary cancer samples, and we can also isolate NK cells from the patients, and then given this as an exosome-induced NK cell. The second question we want to tackle in the cancer microenvironment is that can we generate 3D culture of the microenvironment. So this is another master alumni. She decellularized ovary cancer microenvironment and can produce cancer niche scaffold. Next. Next slide. Okay. When we use NK cells isolated from the patients, we cannot have cell number, NK cell number, over 5,000 cells. We are working with NK cells with the range of 100 cells until 1,000 cells. That's why we turn into regenerative medicine. So in regenerative medicine, the NK cells can be differentiated from CD34 hematopoietic stem cells from umbilical cord blood. So this is showing you that we are differentiating CD34 hematopoietic stem cells into NK cells, and this protocol is also established and has been done by hands-on workshop in all of our master stem cell students. So this is using two sources of stem cells. One is freshly isolated HSE, the other one is cultured for 2 weeks. Next slide. So the next step is, after having discovered we can use HSC as the source of NK cells, and also we can provide microenvironment, we wanted to know whether we can apply the same principle in other type of cancers. And this is another in vitro study result coming from two of our master alumni, showing you that hepatocellular carcinoma also can be regulated by the exosome-induced NK cells. So we take the exosomes now not from the liver biopsy of the cancers, but this time directly from the blood of the patients. So we induce exosomes from the healthy donor serum, and then induce NK cells coming from the hepatocellular carcinoma patient with those exosomes, and we show that several activating receptors are highly expressed, the inhibitory receptors are suppressed, and thus causing the higher capacity to lysis the cancer cells. And this is another finding from another of our master alumni. She showed that we can detect in Indonesia, in the periphery, it's very hard to detect cancers at early stage. So if we can develop a very simple staining using toluidine blue and just stain peripheral blood with toluidine blue, we can show that there is a changes in toluidine blue staining of the cells, NK cells, that endocytose exosomes from the cancer environment. Okay, next slide. Now we are moving this technology in translational, so we are going to transplant the exosome-induced NK cell in a rat model, carcinoma rat model, and this is the work by Nurul Fuad. He is creating a hepatocellular carcinoma rat model, and up to now, I think we have begin with the second batch, third batch, and the different methods to induce hepatocellular carcinoma. Next. Okay, so when I told you in the beginning we don't have CAR-T technology, but in 2024, Professor Alexander McLellan from Otago University, he's an expert in CAR-T, and he come to Faculty of Medicine Universitas Indonesia, make collaboration, and now we are using his plasmid to develop CAR CD19 T cells and CAR HER2 T cells, as well as CAR NK cells from both of these cancer antigens. Yeah, a lot of work. So we have first to isolate, transform E. coli from the plasmid, propagated the CD19 plasmid and HER2 plasmid and all the lentiviral construct. And up to now, I think we are in second year, and we are trying to purify and concentrate the virus to have a good titer so it can lyse the cancer cells. Yeah, next. So as a conclusion of my talk is that our research trajectory in both roadmap, whether it's iPSC liver organoids and cellular immunotherapy for cancer, it's actually never been linear. It's always about what went wrong in the previous study, and we try to improve it better. So for instance, with 3D hanging drop methods, you cannot use more than 2 types of cells. So when we make the primary liver organoids, we cultured, make the cellular ratio, and find the right culture medium into the native liver scaffold, giving some extracellular space on each cells. And when we have made the iPSC liver organoids and transplanted this further, we notice a problem with the transplantation, so we try to make the bioartificial liver, and now moving on to the next generation of Shinta. For the cellular immunotherapy for cancer, starting with the exosome-induced NK cells, making the 3-dimensional scaffold, differentiation of hematopoietic stem cells into NK cells, detection of exosome endocytosis with simple histology staining such as toluidine blue, in vivo study of the exosome-induced NK cells, and now comparing between the CAR technology and the exosome-induced NK cells. With that, I would like to close my presentation with thank you notes. This work cannot be accomplished by all the wonderful talents, the amazing talents, our master students, our doctoral students in biomedical science. I would like also to give acknowledge to our professors, Professor Ismail Hadi Subroto Dilogo, Head of Stem Cell and Tissue Engineering Research Cluster, Professor Shyana Adiwinata Pawitan, our source of inspiration and perseverance, Dr. Amir Mahmud from King Saud University, Prof. Hanriyu from National University of Singapore, Professor Alexander McLellan from Otago University, and last but not least, the upcoming next generation of Shinta. We are having close collaboration with Prof. Nobuyuki Takakura Sensei from Osaka University. Thank you so much.
So thank you, Dr. Radiana, for sharing the research roadmap on liver organoids and cellular immunotherapy in cancer. So our next speaker is Associate Professor Liu Xiao from the Institute of Biopharmaceutical and Health Engineering, Tsinghua Shenzhen International Graduate School. He holds a Ph.D. from the University of Copenhagen and has contributed to a major international genomics projects such as 1000 Genomes Project and the International Cancer Genome Consortium. His research focuses on the human system's immunology in cancer, autoimmunity, and skin disease, with particular interest in translational immunomics for disease diagnosis, progression, and immunotherapy. Today, Professor Xiao will present on the skin microbiome and aging, highlighting the novel insights from multi-omics approaches for targeted anti-aging interventions. So, Professor Xiao, the floor is yours.
Thank you very much for a very nice introduction. So today I'm going to show you some stories our group done in the recent years about skin health. So the topic is skin microbiome and aging. Okay, so So first of all, we are, you know, a lot of us, you know, paying, you know, trillions of dollars each year for cosmetics to prevent skin aging or, you know, alleviate skin aging. So, but, you know, very few, I think not all of us know that skin aging or skin health is not just about the human skin cells, but it's rather a complex ecosystem between skin cells and other, you know, cells and also the skin microbiome. So first of all, if we get to understand about the skin agent, so there's a very important two pathways, two, you know, components. One is the extrinsic and one is the intrinsic. So extrinsic, including the photoagent, that will cause, you know, a lot of pathway, like the degradation of the collagen and also for the apoptosis for the skin cells, and also the skin pathways have been inhibited. And also intrinsic, you know, components like biological agent and other immunological reasons that also will also cause skin aging. Sorry. Okay. So just now I mentioned about the skin health. So it's a quite complex network, you know, among the skin cells and also a lot of the immunological cells within the skin. For example, in epidermis, there's a lot of the, you know, so first of all, beyond the epidermis, we can see a lot of microbiome. And in the epidermis and also in the dermis, there's a lot of different immune cells, like the T cells and B cells, the Langerhans cells, which is a type of the immunological cells, play important roles in human skin, and also the lymph nodes, so very important components that produce inflammatory factors for the immunology of the skin. And also, the skin microbiomes, you know, play a central role in modulating this network between the skin cells and also the immunological cells. So you can see, so one, you know, mechanism is that producing the anti-AMPs and also of the immunological factors like cytokines that have been produced by immune cells for promoting the anti-inflammation and also the recruitment for different immune cells. So in terms of the skin microbiome, first of all, we all know that the commensal bacteria and also fungi, so altogether we call it microbiome, have been, you know, discovered in the human gut, right? We just now mentioned by Professor Xin, and also in a lot of different, you know, human organs like oral microbiome. But skin is also very large habitat, large places that with a lot of commensal bacteria. The diversity of the skin microbiome is quite complex. It's even more diversified than we anticipated. The study about the skin microbiome have increased a lot in recent years. So there's different roles that played by the skin. First of all is the like physical barrier. That, for example, we can see the important cells beyond the epidermis, and also some of the other barriers, including dermis and the epidermis. So and also the skin bacteria have been playing a major role, have quite causality roles for inducing some of the skin-related diseases. For example, for the acne, which is quite often, happens in the younger generation, the acne. So we have already discovered that certain strains of the C. acne, which is a common type of skin bacteria, have played a major role in inducing the acne. And also for atopic dermatitis, which is also very important, very serious inflammatory skin diseases in the human. So we have already know that S. aureus, especially some of the specific strains that producing the damage factors that for this S. aureus have been played a major role, quite correlated with the AD, we call it atopic dermatitis. And also for other skin-related disease like psoriasis, like the lupus, have been all discovered have a causal relationship with the human, with the skin bacteria. So before we get into study the correlation or causality between the skin microbiome and also the skin health, first we have to understand what's the composition of the skin microbiome within the human skin. So now, before our study, so this is work we studied several years ago. So before we study, because we know that the skin microbiome is a very tiny, component for the skin. So it's very difficult to enrich this bioactive component and to study the diversity for it. So previously, so scientists mostly using the new 16S, sequencing technique to study majorly the species within the human skin. But we don't know yet the functionality for different genes that of the skin microbiome. So we have developed techniques that can enrich and amplify the very tiny components of human skin microbiome to study the gene function for different species of the human microbiome, the skin microbiome. So we get to, this study, we extracted the samples from the facial skin, including nose, including the cheek, and we get to understand the different components, including the bacterial, the fungi, and also virus. That's the major components in the human skin, the facial skin. So we get to know that the skin is still very important, several important, the bacteria, including C. acne, including S. aureus, which have been recovered before, that have been enriched in human skin. And a lot of other fungi and virus have been also discovered. So then we try to study so this is a cohort study. So we use this technique, for first time, we've been able to study the composition in a cohort. So this is more than 200 individuals. Okay. So, so then we try to understand the function of the skin microbiome. And we do a pathway enrichment. We found that a lot of the important functions have been produced by the skin microbiome, which can compensate the human cell function. And another interesting phenomenon, so we identify a lot of the anti-resistance genes in the human microbiome. That's consequences of the human using the antibiotics. Okay, so then we try to, because we're doing a cohort study, we have a lot of individuals, so we try to classify the human skin, using the skin microbiomes. So for first time, we identified two we so-called chorotypes in the human skin microbiome. So one chorotype is mostly related to the, to younger skin. So it's with a lighter skin, skin color, and more hydration, and also the darker, and also the more porphyry. So this is what called the C chorotypes. Another chorotypes, we call it M chorotypes because it's mostly enriched by the M bacteria, which is Osteomyxis. So these bacteria have been enriched in M chorotypes. And these chorotypes have been found majorly in aged skin and also enriched with the low hydration and the darker skin color and low porphyry. So a very important question we want to ask is that we already know that the skin bacteria can induce the skin-related diseases, but whether the skin bacteria can induce skin health, skin aging is something we don't know yet. So first of all, that if we can identify some specific bacteria that's related to or can cause the skin aging, so we can to target them as a potential therapeutic or intervene ways to do the skin health intervention. So first of all, using our cohort data, we identified a lot of the specific characteristics of skin microbiome have been related to the chronological aging. So we can see that the skin bacteria contributes the most to the I'm sorry. So a little bit slow. Sorry. Okay. So you can see that this figure shows us the contributions for different skin phenotypes, including the bacterials, the different bacteria contributes. So we can see for the specific agent that bacteria contributes the most. And then comes the fungi, and then comes the virus. So we identified as previous study that the diversity increased, especially the better diversity of skin bacteria with the chronicle age. And then it's a bit slow. Okay. So and then we try to identify specific skin bacteria that's related to chronological age. So we identified one bacteria, which is the C. acri. We previously know that is quite related to acri, that's being enriched in younger skin and decreased with the chronological age. And another skin, which is the Moraxi osnonesis, so it's another M bacterial, have been enriched in a older skin and increased its abundance with the chronological age. So this is the first time we identified for the correlation with the human skin agent. So we also, besides the correlation analysis, we also built a machine learning, which is called a skin agent biological clock. So using this biological agent clock, we also identify similar results that C. acri in being reached in the younger, skin, and M bacteria being reached in the older skin. So using these two approaches together, we identified that the important roles for the M bacterial. So another question we want to ask, whether this correlation is just a correlation, for example, because of the changes for the skin microenvironment of the older chronicle aging process, for example, the nutrition that cause the changes of skin bacteria, or it can also be a causality that the skin bacteria can induce the skin aging phenotypes. So this is a question we want to answer. So using some of the experimental approaches, for example, we are first neutral these specific bacteria, for example, M bacterial, and we use the supernatant, to treat the important dermis, for example, fibroblast skin cells. And we identify some of the important pathway changes that this bacteria can induce. For example, we can see that the M bacterial can induce the upregulation for MMP proteins or genes. That MMP is very important for degradation of collagen in the human skin. So then, the induction of the M bacteria can induce the degradation of the collagen in human skin and then promote the skin aging. So this is a phenomenon. This is the hypothesis we raised for M bacterial. But the most important thing we want to answer is that because that if we in the same chronological age, but we show very different, skin aging phenotypes. So some people look very young and others look very old. So we want to know whether skin bacteria plays some roles in this process. So then we do another cohort study that using a large number, so more than 100 patients. So this study has been, done by our Ph.D. student, Guodani is also here in a meeting room. So this study, we wrote more than 100, individuals that's within the same chronological age. So mostly they are around 40, so between 39 to 41. So why we choose 40 years old? Because in these years, so the people, especially women, so is getting the waterfall for the skin aging. Some people, some women get very, quickly accelerate, skin aging. Some, can retain or even, anti-aging. So we choose this age group. And we choose, 50 women that with a very prevalent, younger skin age, and another with a very prevalent older skin age. We use the, AI approach to look into the phenotypes for skin aging, and also use elasticity meters to, to measure the elasticity, because elasticity is the most important skin aging, characteristics. And then we, separate the skin aging, groups into two groups. One's the younger group and this is the older group. And when compare the skin microbiome, especially skin bacteria, and also together with the metabolites. So we got very important, very interesting, discoveries. So first of all, as we, can anticipate it, that a lot of the skin characteristics have been very well differentiated between these two groups. For example, the porphyrin, like the elasticity, of course, we use it as the parameter, and also the skin color, and also some other, very differentiated skin characteristics. And besides that, okay, So besides that, we identified a lot of the very interesting bacteria, specific bacteria have been related to the skin aging phenotypes. So for example, in the younger skin, we identified some of the auras, some of the bacteria, two major bacteria. This Humanus auras is another kind of the string auras. And especially in the younger skin, we identified a lot of the bacteria, big bacteria. The one important bacteria we identified, is shown here. So it's the one we want to study. But the interesting part is that previously, and as we showed here, and also other publications we show that C. acne have been enriched in the younger skin, but we don't identify it in our study. So why is that? We want to answer the question. Is it related to the cohort selection or related to something? So we hypothesize that the enrich or the more diversity for the older skin, right, is quite related to the C. acne. Okay, so want to emphasize that. So in previous study, and also in our previous study, we identified that the younger generation have a lower beta diversity, the skin bacterial beta diversity, than the older skin. But this study shows the opposite, that the younger generation is the more diversified, and the older generation has the less diversity. So why is that? Why is this so different? So our hypothesis is that the enrichment of C. acne have played a major role here. So because the C. acne, is very abundant in the younger generation, so it cause, the higher diversity for the younger skin. So if we, deprive, to get rid of the C. acne bacteria, then we measure diversity for the younger and older skin. So we can see that, as we anticipated, that the older skin have a lower diversity and the younger skin have higher diversity. So then we want to identify some of the very important metabolites that's related to the skin bacteria. So some of the metabolites have been identified as for example, melanin, that's related to the skin color, and also some of the important pathways have been produced by skin bacteria have been identified by our study. Then we use the GM analysis, the genome-wide metabolic network analysis. We want to identify these bacteria, which is called S. multifida, which is the most enriched in younger skin. Why it has a phenomenon? Why it has correlation with the younger skin? So we use this GM analysis to identify that this specific bacteria that can convert the oxidized GSH to the reduced type of GSH. So it's involved in the anti-oxidation for the skin metabolism. That is a potential mechanism for the anti-skin. So then again, we want to study whether it's a correlation or causality. So we did an experiment. We used the superlatant for this S. multifida, the specific bacteria, and we used the superlatant to treat the fibroblast of the skin cells. And first of all, we built an aging model or oxidized aging model. We used the oxidized to treat, to induce the aging skin model. And we found that the specific bacteria can indeed induce some of the aging pathways. Like, for example, for the model we built here, we can see a lot of the cell synthesis and also the skin aging, regulation for skin gene pathways. And after treating with the superlatant for the specific bacteria, we can see that the aging process happened. Anti-aging process happened. And we also look into the phenotypes, for example, for our hypothesis that these superlatants can induce the reduced type of GSH. So we measure the oxidized GSH and with the reduced GSH, and it is the same as we anticipated. And also measure the aged skin cells. Using the study, we found that the superlatant can really reduce the aged skin cells. Okay, so besides this study, we identify specific skin bacteria that has a causality relationship with the skin aging. We also, want to understand it because, currently a lot of the young ladies using the retinol, within the components for the anti-skin aging, right? I don't know whether you have used it, but it's very popular in China and also in the United States and other countries. So retinol we already know that retinol can reduce the skin aging by, regulating the skin cells. But whether this process have a correlation relationship with the skin microbiome is still under investigated. So we also designed a study that we recruit, young ladies that using this retinol for cosmetics, and we study the dynamics for a different stage of the using the retinol. So we can identify a lot of the, skin phenotypes have indeed been, induced by the retinol. For example, some of the younger, skin phenotypes like the elasticity, like the color and the porphyrin have really been induced by the retinol, as we anticipated. And we also identify some of the important skin bacteria having dynamic changes in this process. And we also identify a lot of the metabolites that related to the bacteria having dynamic changes in this process. And using GM analysis, we found that, for the first time we found that the skin microbiome, especially the specific skin bacteria, have been involved in the process of a retinol, retinol metabolism. So this may be one of the mechanisms that the skin microbiome have been involved in the retinol anti-aging process. So this study afterward posted in the bioarchive. So the Utah scientists very interested in the study and write a report for it. Okay, so if we identified specific bacteria, for example, M bacteria have been related to the skin aging process. So then how to modulate it, how to intervene it. So one possible solution is the MP, antibacterial peptide. So we want to use the MP as the potential therapeutic strategies or intervening strategies to inhibit specific bacteria that's related to skin aging, but not, degrade, not, to disrupt the skin microbiome hormone status. So this is called the targeted MP. So we think that the skin microbiome could be a very rich resource for a targeted MP. So this study has been done by our master student, Huang Yixuan, also in this meeting room. So he designed a pipeline using deep learning to identify, to enrich, and to classify potential AMPs in a specific, for example, human skin, bacterial sequences. And using this using this pipeline, we identified a lot of the AMPs potentially have a therapeutic, effects. So this, we are using we are synthesizing some of the AMPs we discovered. For example, this is MP3, and we found that indeed they are specific targeted our anticipated bacteria. So this time in this study, we want to decrease, we want to inhibit the SORs because we want to treat the AD, atopic dermatitis. So we use the MP3 that can specifically target the SORs, but not, to interrupt the other, skin bacteria. So it can maintain the skin microbiome hormone status. So using this MP, so we not only found this inhibition, but we also use it to treat the skin model, three-dimensional skin model. So this is done by our PhD student, Ya, who is also in this meeting room. So she designed also, to use the three-dimensional skin cells to construct the AD model and also to add the SORs into the skin AD model. We found that the specific MP, our targeted MP, can reduce the inflammation for this AD skin model, but also can reconstruct the structure for the skin types, and also can reduce the SOR's abundance. So this is as we anticipated. So this is the conclusion. So first of all, we found that the skin agent, is quite associated with the skin microbiome. And we found a lot of specific, bacteria like M. bacteria and S. Mountifinia that not only has a correlation with the skin chronological age, but also have a correlation with the skin agent phenotypes. And not only the correlation, but it also has the causality effect for the skin agent. And we also identified that the skin commensal bacteria are involved in a skin retinol, this is an anti-aging, component, skin retinol metabolism process. And we can target, this specific, bacteria by the AMP, by our targeted AMP to intervene the skin aging. That's a potential, solution we want to do, but it's still in a very early process. So this is stories we want to share with you. And the next, I want to use several, a couple minutes, just 3 slides. Okay, so first of all, it's an acknowledgement without their help. So including Guo Danli and Wu Yahong and also Huang Yixuan, they are in this meeting room together and here. And so our study cannot be facilitated. So next, I will use the 3 slides to introduce some of the, our iBHE program. So I can see a lot of students here, so I want to show you. Just now that Professor Xing have shown introduction about iBHE. I want to show you some of our programs here. So in case that, in case you are interested, that we, in iBHE country, we have 5 different PhD programs and 4 master programs. That if you have you have a master degree, you can apply for a PhD. And for our master programs, we have the precision medicine, which is the all-English program, and also we have the biomedical engineering, and we also have the pharmaceutical engineering and health evidence-based health engineering. So these 4 master programs, so they offer both English program and also the Chinese program. So you don't worry if you don't understand Chinese. And we also have different PhD programs. If you're interested, you can apply. And this is very slow. So this is about some of the application guidelines that we are showed here. So you can see our application system have opened for each applicant. We are doing several rounds of the interview. It's mostly the online, not face-to-face, but online interview. And you can scan this code and to get more information about the application process, about the application materials, and all the other things. And for those you want to apply for iBHE, so I think that there's a lot of support. So this can see that we are providing a lot of the assistantship and scholarship to compensate, to cover the tuition fees, the accommodation fees. The accommodation fees is very small, so don't worry about that. So I think the school will cover for that. And we provide different kinds of the scholarships that can cover most of the tuition fees. And also in iBHE, we will also provide the student mentorship for each of the international students to help you solve all the problems regarding your life in campus, on campus, and your life in study. So don't worry about anything. We have a mentorship for you. So this is things I want to introduce to you. Thank you for listening. That's all for today. Yeah.
So thank you, Professor Xiao. So I invite all of the 3 speakers to come forward so we can discuss with the participants. Professor Xing, Professor Radiana. Yeah, so such a 3 interesting topics. So I invite all of you who wants to ask questions, and then we have also questions from the online, so I will read the questions. Yeah, Dr. Rizky. Introduce yourself first.
Okay, I think it is not morning, right? So I have good afternoon, morning to afternoon for distinguished speaker, Prof. Xin-Hui and Associate Prof. Liu Xiao and Dr. Radiana. My name is Muhammad Rizky Fadila, so called Rizky. And actually, I really take some lot points, but one thing that has been stated by Prof. Xin-Hui, since it is related with my great research about drug discovery. Obviously, we have see your presentation about how we develop peptide-based drug. It is also one of pipeline that have been done along with my dissertation. But my question is, how Tsinghua University, especially maybe from your lab, Prof, translate the finding? I mean, when you find those lead compound, ideal compound that have a specific mechanism, one of which is DPP inhibitor, how you could reach those mechanisms to the patients, especially in China. Maybe I missed the point, but could you please tell me about that again, Prof? Thank you very much.
Yeah, thank you, Dr. Rizky. The translation of the discovery of peptides, Prof.
Yeah. Thank you for a very nice question. I think basically the target is from the human. So if you identify the natural bioactive peptide, I think basically it's the safety can be made sure. So, but currently we just use the cell model and also the human organoid model, combining with the animal model. But currently, I think in China, if we develop the functional food by combining the new peptide inside the product, so that proof will be enough, evidence will be enough. But if we want to go to the such as the so-called the hospital applicable peptide products, in that case we have to do the clinical test. But clinical test basically not like the peptide drug, not so phase 1, phase 2, phase 3. Just only the very limited the patient will be enough. So which is we are now cooperating with nutrition, the department of the hospital. So that I think that in future we will extend our application to the hospital, the nutrition foods, the peptide food. In that case, we will do the clinical test, but currently we just focusing on the functional food. That's enough. In another sense, because all the peptides we have discovered, I think there's a very, very new structure. There's no the previous publication reported. So in that case, it's basically it's a very, very good candidate for developing the peptide new drugs. In that case, we have to follow all the process. But currently we have using the website technology. So we estimated the drug ability. So all the peptides, especially small peptides, less than the 10 amino acids, in that case, all the peptides are very stable. So in that sense, it's easy to develop the oral administration, the peptide drug in the future. So we are cooperating with a big company in China, so to translate all VMP to the, I hope in the future can be a good target for the peptide drug, for the anti-diabetes, the new peptide drug, I hope. Thank you. Yeah, sure. I think they just demonstrate that the AI plus the computation plus the molecular docking, I think is a good way to accelerate the discovery speed of the new peptide. Yeah, thank you.
AI.
Oh yeah. I didn't mention today, but we also discovered many novel peptide, bioactive peptides from the aquarium animals. They farmed aquarium animals. Also, they're very unique. Yeah.
Yeah, thank you, Prof. Xing. Yes, so it's a lot shorter, yeah, to develop than the conventional pharmaceutical medicine. Yeah, thank you, Dr. Rizky. Any other questions from the floor? So, yeah, please introduce yourself first.
Good afternoon, everyone. My name is Adria. I'm really interested in from the Professor Liu Xiao research, and I want to ask that we know that skin aging is actually inevitable process, but to what extent that we can modulate the skin microbiome to optimize the anti-aging strategies? Thank you.
Thank you, Professor. Very important.
Very important question, and also very open question. I think we don't have the answer yet. Yeah, because but we already know that by using iteration. For example, you believe that, right? By iteration, by using different cosmetics, by using different skin health product, that you can to get a better skin age, better look of the skin. So our hope is that by identifying potential components that within the skin bacteria, that all some of the molecules that can inhibit, for example, AMP we just mentioned, are still in a very early age. So for these bioactive components or products, we can add into the skin health products that can alleviate the skin aging process. So that's our hope. But to what extent that process can happen, we don't know yet. So let's see. Okay. Thank you.
Okay. I hope that answers your questions. Yeah. Thank you, Professor Xiao. So I can invite maybe one more questions. Yeah. So
Okay.
Yeah.
Thank you for the presentation. My name is Salisu, a master student for the master's program in biomedical science. My question is to the first presenter, Professor Sir. You highlighted the project you are working on and anti-diabetics. Is it only on diabetics that you are working, or there are other investigations you are doing at your center? Thank you.
Sorry, I didn't get your point.
The anti-diabetics?
Yeah, you highlighted your project on anti-diabetics
Your
using
project on
peptides.
anti-diabetics.
So is it only diabetics that you are working on, or there are other disease processes you are also investigating? I'm asking whether you are doing other projects.
So you mean which state?
Other indications, maybe, Prof. Other than anti-diabetics, maybe you are working on also on other.
Oh yeah. Yeah. So today I just showed one research programs, but we also are developing other. They are maybe dipeptide, 2 amino acids, and maybe several the amino acids also have the DP4, the inhibition activity. So that also is a good combination, a good way for developing the anti-diabetes. I think the health food, the functional food, but maybe the key strategy that has a very high activity with a lower IC50. In that case, there will be also possible in future to develop new novel peptide drug for treating the diabetes. I think that's the one stone two birds. The research approach we are doing.
Thank you, Professor Xing. And then in relation to Salisu's questions, I have one question from the online participant, from Dr. Supraja Dwiyono. He wants to ask about the drug FAMP1, Professor Xing. Since it controls blood glucose and body weight, can we drink or consume it for the healthy people to aim for lower blood glucose after eating high glucose food, or maybe lowering body weight, because it's related.
Yeah, for the industrial head, I think their products will be good for the healthy people to prevent such as the weight or the weight increase, or to control the glucose concentration, the level. I think that we already have some different the animal, the evidence. Yeah.
Okay.
Also myself.
Okay, so Professor Sisi also consume it, yeah. So and then before Professor also has already shared the paper about it, and then I will give it to the committee. Maybe the committee can share it to the. Is it okay, Prof, to share it with the participants online? Your papers that you just.
Oh
shared,
yeah, no problem. That paper is already published.
Yeah.
Please.
Yeah, thank you, Professor. And then maybe the last questions I want to ask Dr. Radiana in regards with your IPS liver organoids that you have been working in for years, right? Yeah. So how is actually the biggest obstacles from your experimental models for your clinical translations? Yeah.
Okay. Actually, I wanted also to ask Professor Xing and Professor Xiao on what they think about the research that we've made here, because obviously it's really amazing to see intelligent data and a vast amount of data shown from single transcriptomic levels into peptide omics level. So I think if we have access to those facilities, of course our research, in vivo research, our translational research with the animal models can be presented better and will not have such a discontinued array of data. So I think that the biggest challenge is that, Professor Melva, when we do research and we only display like histology, stainings, immunofluorescence, electron microphotography as disparate or discontinued data, sometimes it's very difficult for us to have intelligent discussion on the mechanism of action. And I think if we can collaborate more on that part, maybe it will help. Maybe I would like to also ask your comments on that.
Yeah, maybe Professor Xing can comment on the liver organ.
I think for both Indonesia and China, okay, I think the translation is a big task, but translation is a very complex process. So firstly, I think in China case, how to solve the IP issue. But IP now, the Tsinghua has established a very good the protocol to translate the IP to industry. So in many cases, the company or the industry, they kind of solve the very, very advanced new technology. So this, I think in China case is some sense is minus the effect on the translation. But in that case, even all students or your student in University of Indonesia have their their ambition to do their entrepreneurship. I think that's a very, very good way. Personally, I have 4 technologies successfully translated into the industry. The 3 of the technology have been translated by my former Ph.D. students, they have the entrepreneurship spirit. So now successfully translated into the product. But that's I think if the PI or professors don't have this interest, I have the interest. So also in Shenzhen, we always say if their research result achievement cannot be translated, I think that's the result for Shenzhen government. Because Tsinghua has been strongly supported by the Shenzhen government in Shenzhen campus. So the responsibility we have to do the translation. But now in Shenzhen, I think relatively it's easy to do the cultivation because all the industry, the people have their strong, intensive, the insight to solve the new technology, to the new cultivation. I think that's a very good one.
Yeah, yes. So I think from today's lecture, I can see there's a lot of the collaboration opportunities between us. So though you only have one speech from the UI side, right? But I can see from your speech, you're talking about the liver organoid for the tissue regeneration, for tissue transplant, right? For liver transplant, for cirrhosis, and you're also talking about cellular immunotherapy. Right, I think that for the liver organoid, I think Professor Xing is also doing a lot for liver organoid construction using iPSC, right? But he's not for the tissue generation. It's not for transplant, but for by assay, for testing the drugs, right? Testing drugs, but also similar things, I think. And also for the tissue regeneration, we have some, professors in iBHE that are doing that, right? By adding some of the components, some of the, to make the tissue more affordable, more, storage better, and those things, I think the potential. And also for the immunotherapy, I think that this afternoon my students, my PhD, are also talking about something about that. We are also very interested in T-cell immunotherapy. Yeah, but not CAR T, but we are looking for, using the T-cell receptor therapy, especially not only the T-cell receptor T-cells, but also the protein drugs. So that's something that Mondanda is currently, working on it. So I think the great potential opportunities between us. Okay, thank you.
I think in China, as you mentioned, their CAR T or their organoid really also is a very hot research area. Especially maybe 2 weeks ago, Chinese government has approved the clinical translation of their CGT, the cell and gene therapy, for clinical speed up their clinical process, as signed by their Prime Minister, of Liu Xiao. So I think that's a very good driving force for the industry translation. But I think the case, as you mentioned today, one of my questions is how in Indonesia, how do you evaluate the biosafety of the iPSC-based transplantation, organ transplantation in the future? This is one question. The one comment I just say quickly. In Shenzhen, we have many associations. At least two associations we are jointly collaborating now related to the cell therapy or the gene therapy. So I think in the future, in Shenzhen and in your university, I think we can make a strong pipeline for the collaboration in this area too. Okay.
Thank you, Prof. A short answer. So yeah, for the iPSC, that's why we put it outside the body and make a bioartificial liver. But I mean, in gold standard, we have to inject in non-skid mice to see the teratoma formation. Yes, safety issue with iPSC is a problem. Yeah.
Okay. Thank you very much. Yeah. So because we have a very limited time, so with a very deep thanks to our three speakers, Professor Xing, Professor Radiana, Professor Xiao. So I'm thanking all the three outstanding insights on the synthetic biology, regenerative medicine, and then skin microbiome. So with this, I conclude the study. So I give the time back to the MC. Thank you very much.
Thank you.
Thank you very much, Prof. Melva, the speakers, and also the participants. We would like to take a moment to express our deep appreciation to our moderator and distinguished speakers for their insightful presentations and valuable contributions. Now, we would like to kindly invite Prof. Xing, Associate Prof. Dr. Radiana, Associate Prof. Liu Xiao, Prof. Melva to come forward to the stage once again. The souvenirs and tokens of appreciation from the Faculty of Medicine Universitas Indonesia will be presented by Prof. Wawaimuli, Head of the Master's Program in Biomedical Science, Faculty of Medicine Universitas Indonesia. We kindly ask for the committee to assist in organizing the photo session. Okay, for everyone, please. Okay, now we want to take a picture of us. So the professor will be seated in the front of. Okay. Ya, kami itu di depan. Kak, ke tengah. Ke tengah.
Boleh menuju diisi yang kosong. Biar bagus.
I will give instruction 1, 2, and 3, and then cheese. Okay, ready? Okay, let's begin. 1, 2, 3. Say cheese. Cheese. Other pose, other pose. Oh. With Xinghua flag. Okay, ready? 1, 2, 3. Oh, say cheese. 1, 2, 3. Say cheese. Cheese. Other pose. Okay. Thank you very much. Ladies and gentlemen, please be seated. We have now reached the lunch break session. The lunch and cultural performance will take place in Auditorium 2, located just next to this room. During this session, you are invited to enjoy a special traditional dance medley of Nusantara, performed by Sanggar Art Nusantara Production, proudly presented as part of today's insightful program. Please proceed to Auditorium 2 to enjoy the performance and lunch. After the break, at 2 p.m.,